Okay, so here is topic 10. We're going to be talking about the cell cycle. So this is just overview of cell cycle. We're going to start. First off, there is three types of, well, not three types, but they categorize it in three ways or three types, three reasons that they cell divide. One's for asexual reproduction, so those are unicellular organisms that divide for them to reproduce. There's growth and development of embryos. So this is all about how they become the mature species or to become the mature organism from embryo. It's not really about gametes or haploids. It's really just about growing, but it's the differentiation between growing and repairing. Sort of repairing is like renewal somatic cells. So just this is for continuing to live on, whereas this is for maturing kind of. And of course, there's haploid, diploid cells, and there is the mitosis and meiosis, which we'll talk about. But either way, they are part of the cell cycle, and there is the meiosis and mitosis. And yeah, those are the types of cells. So there are somatic cells, body cells, and gametes. Gametes are haploid or germline cells such as sperm or eggs, and somatic cells are for your body cells, basically everything other than the sex cells. Yeah, just note that mitosis are identical cells after they're created, and the meiosis are individual. They give rise to the gametes that are pretty much independent. Well, they're not copies. They're not clones. So then now we have DNA and chromatin and chromosomes. So first off, chromosomes is right here, are genes. Well, they have genes. They contain genes that are specific, and these are all hereditary information from either your mother or your father, and we'll talk about how they could be homologous from one of your parents. And they are coding for primary protein structure. So chromosomes, DNA, and genes are coding lots of structure of protein information. And the bundle of chromosomes, like the collection of all chromosomes, is called a genome. Genome is a collection of all chromosomes. There are 23 in the human body, a total of 46 chromosomes. So 23 different chromosomes. There are 23 pairs, and there are 46 in total. Chromatins are the when they're bounded with proteins, positively charged proteins that act as spoles that we call. So it's usually histones, but these are positively charged proteins. They were called, yeah, it's just proteins. So there are 4 pieces, or I should say 8 pieces of histones, protein complex. So DNA plus protein is what gives chromatin. So DNA plus protein, chromatin. So when they condense, they will make the DNA. When they condense, so when histones condense, well, they will become heterochromatin instead of euchromatin. So euchromatin is when it's loose and allow expression of genes. Heterochromatin is when it's compact and its nuclear membranes are it's close to the nuclear membrane. They are dark and you can see them through microscopes when they're in heterochromatin, and they are silenced mostly. The genes are silenced. That's just my timer. Okay, so they're tightly compact visible, and yeah, that's the difference between euchromatin and heterochromatin and euchromatins are when cells are not dividing. Yeah, so they can condense. So chromatins will condense to become chromosomes. Chromosome numbers are 23, so 23 pair, one from dad, one from mom. There are 22 that are autosomal, so meaning these are these bigger ones, and there are 1 pair of sex chromosome. We say that they're pairs, although they're not really identical pair. They're not exactly homologous, but we say that they're homologous during meiosis because they can, in some ways, do the crossing over, but we don't get into that detail. So 1 complete set is a haploid. 1 complete set is haploid. 2 complete set is diploid, meaning if there's one that comes from another parent, so if there's like a set that's from different parents, that's definitely haploid. Diploid. If it is only from one parent, then that's haploid. So 2N and N. 2N for diploid. They come from paternal and maternal. So karyotype is this guy right here. It's the graph that we see like this. Not a graph, but it's a representation. We call that a karyotype. And you can see the different chromosomes. Homologous chromosomes. Homologous are these 2 parents, so these chromosomes have the same genes but different alleles. I went to French school. Alleles, and they are alternative version of a gene. So there are alternative different versions of a gene for the same appearance or behavior characteristics. So they work for the same gene, but the alleles are different, so from different parents, basically. So one could be for coding for a blue eye, the other one for a green eye, etc. So the cell cycle is all about dividing to two new daughter cells. So that is for mitosis. Yeah, so when it divides to two cells. Yeah, so let's go into the cell cycle, formation of cycle, two new daughter cells, copy of genetic information, and let's get into interphase already. So there is the interphase and the mitotic phase, which we'll talk about in the next capsule. Interphase is about G1 synthesis phase and G2. So there are 3 steps in the interphase. And the most of the cell's lifespan, around 95%, is the interphase, and very little bit is mitotic phase. So around 5% is mitotic phase, which is the mitosis. And yeah, interphase is about 10 to 14 hours long. They are most actively in multiplying mammalian cells. So G1 phase is for organism synthesis. There's an increase in size of the cell. Everything is synthesized from organelles, and they reach the normal mature size, I should say, just increase in size, and they are synthesizing regulatory proteins. Important proteins are synthesized in G1. So G1, the first growth size is when the proteins are regulated. So these proteins are later on going to be important because they're going to be check marks for each stages. Interphase S phase is also very, very important. It's basically the biggest thing we should know about the interphase, because they are the one that replicate DNA, and they make from one chromosome to two sister chromatids. That's literally the most important thing about this phase. They contain the same genetic information, so these two contain the same exactly identical information. So sister chromatids are exactly identical, and there is a centromere that attach them. This is half the cell cycle. Yep, so exact same copies are made. So this is duplicates. The duplication is what's happening here. These are called sister chromatids, lined by centromeres, not centrosomes. So these are homologous, and these are still homologous chromosomes. Even if they're doubled, they're still called chromosomes. So G2 phase is the final stage of the growth. So the second stage of growth, they will increase in size as well, and they're going to be preparing for mitotic phase. So this is like the preparation growth phase for mitotic phase, and the centrosomes are duplicated. So important to note, G2 phase of the interphase, centrosomes are duplicated, and spindles, fibers will form to make them separate from each other. I should have drawn a picture. So basically these are the centrosomes. They duplicate, and after they duplicate, they will try to make spindles so that they separate from each other, and we will go into that later on as well. Classification. So this is a clarification. What is a chromosome and chromatin and chromo etc. So chromosomes is the linear, and there's different for each different gene. They are linear, just chromosomes that we see over here. So all the bundle of DNA like this, and that is the condensed form of chromatin. Chromatin is when it is not forming chromosomes. So chromatin will turn into chromosome when they are going to cell divide. They are loose. There's euchromatin and heterochromatin, and they are DNA and protein complex. They will condense to become chromosomes. Chromatids are when the chromosome divides. So it's like this. It's like this kind of trajectory. Condense to become chromosome duplicate to become chromatids. Chromatid sisters are done in S phase, like I said, and they duplicate of the exact identical chromosomes, and they're still called homologous. This one and this one is homologous. They're held by the centromere, and they're called sister chromatids. And that's it.